Okay, so last time we were talking about the location of the different lobes of the brain, and most of them they have their name based on the bone that they are in contact with the exception of this lobe that is deep between the frontal lobe and the temporal lobe, which is called the insula. But the rest are superficial, and as you can see, they have different names. So this is the anterior view of the brain, posterior view, anterior region, posterior region. This is the lateral view. So what you can see in this lateral view of the brain, it is the frontal lobe anteriorly, occipital lobe posteriorly, and then superiorly we have here between the frontal and the occipital, we have the parietal lobe, and then below the parietal lobe we have the temporal. And now, if you see this same view but without retracting the temporal from the parietal, what you can see is that we have here a fissure that is called the lateral fissure, and then we have a sulcus here, which is similar to a fissure but it's less deep, that separates the frontal from the parietal. And now, the gyri, which is basically a ridge of tissue between, ahead in this case of the central sulcus, this gyrus is called the precentral gyrus, and this one behind the central sulcus is called the postcentral gyrus. So a gyrus is a ridge, a sulcus it's a little indentation, and then something that we do here in the precentral gyrus is send commands with the neurons for muscles to contract, the skeletal muscles. So this is what we call the primary motor cortex. But before that, we plan the movement here in the prefrontal cortex, in the frontal lobe. So we plan the movement here, we send the command to this area so that we can start moving our muscles. Our left side of our brain controls the right side of our body and vice versa when it comes to somatic movement or movement of our skeletal muscles. And then here in the postcentral gyrus, we have this area that it is receiving a stimuli from sensory information. So this is called the primary somatosensory cortex. So sensations arrive into this area. So if someone pinches you the arm, you can feel it basically in one of these regions in here. And any sensation like hot, cold, sensory information is here. So motor, precentral, sensory, postcentral. And then here, occipital lobe, it is specialized for visual information processing. And then temporal lobe, it is important for hearing equilibrium and also for memory. And now this is the superior view of our brain, showing you the hemispheres, so left, right, separated by this longitudinal fissure. And then you can still see all the lobes. So this is anterior, this is posterior. So here we have occipital, frontal, parietal, temporal just below there. And then you can see the sulcus. So that's the anatomy of the brain. And now for the encephalon. The encephalon has region and functions. So it has the thalamus region, which is the one that I told you that relays the information into the particular area where it's needed, if that information is needed. Whatever you don't want to be aware of, you keep it in the thalamus, apparently. I guess you can still override part of that information. So it's like the relay center. And then we have the hypothalamus below the thalamus that has several nuclei with different function. We will see just a few of them, not everyone because it's a lot. But at least in that area, we control the body temperature, we control sensation of hunger, thirst, and other drives. And then above the thalamus and anterior to the thalamus, we have a small region that's called the epithalamus. So in this region, actually it's posterior, not anterior. In this region, we have a tiny gland that is called the pineal gland. So this pineal gland is specialized to secrete melatonin. I bet many of you have heard about this hormone before, melatonin. What does people, or why do people take melatonin pills? To help to sleep. So this hormone keep us, the hormone help us to fall asleep during night, and then we control the circadian rhythm in here, so we're supposed to be awake during the day and fall asleep during night. And then we have the subthalamus, which is a region that it is specialized to control motor functions together with basal nuclei, which includes the putamen, globus pallidus, the red nucleus. So here are the different regions of the diencephalon. This is a sagittal cut of the brain, so you're looking internally, the mid-sagittal cut. So here we have the diencephalon. This part is the thalamus, the relay station. And then in here we have the epithalamus with the pineal gland, the one that secretes melatonin to help us sleep during night. And when we don't secrete as much, we have sleeping problems. And then underneath here, we have the hypothalamus with a lot of nuclei that controls thirst, hunger, etc. And then it is not labeling here, but just below this region in the hypothalamus, we have a tiny gland that is called the pituitary gland, which is the master gland. That master gland produces many hormones that controls many functions in our body, including growth in height, because we secrete that growth hormone, metabolism, because it sends a hormone that is called TSH, thyroid stimulating hormone, that stimulates our thyroid gland in our neck to produce thyroxine and other hormones that controls the metabolism in our body. So then those are the regions of the diencephalon. And then this is just a three-dimensional view of this diencephalon, the thalamus, epithalamus with the pineal gland, hypothalamus in here, and here we have the pituitary gland and the subthalamus in here. So how do we secrete melatonin? Well, we have visual input to secrete melatonin. Our eyes detect the intensity of light, and as the intensity of light reduces during the day, we send a command into this region in the thalamus, the suprachiasmatic nucleus, or SCN, that relays information into a part of the brainstem, which is called the reticular formation, that basically sends information back into this epithalamus so that we can secrete melatonin, and then we have information, negative feedback, so that we have less stimulus of our cerebral cortex so that we can fall asleep. So that's supposed to decrease our activity in our cerebral cortex. there is a lot of controversy in relationship whether blue light emitting from our electronic devices can decrease the secretion of melatonin or not. Of course, whoever sells these glasses that filters that blue light will say, yeah, blue light is bad for you, and you have to buy them so that you can still use late at night your electronic devices, and then you don't think that it's still day, and then you fall asleep much better. But, I mean, there have been studies where that is not shown, that you have decrease of melatonin secretion. So in general, it's advisable, guys, blue light or not blue light, that at least 30 minutes before it's our regular time to fall asleep or to go to bed, to stop using our electronic devices. Otherwise, we'll have problems of sleep. Okay? Except during assignment time. In there, yeah, you can still use your electronic devices. Okay, now let's move into looking at the functions of the cerebellum. So the functions of the cerebellum will be to coordinate voluntary movements of our muscles and also to reduce motor error. When we send that information from the prefrontal cortex into the motor cortex to execute the movements, we send a lot of information. And then if we didn't have the feedback from the cerebellum, our movements will be gross. So we will have a bunch of error. So then, with feedback between the cerebellum, the cerebral cortex, the basal nuclei, we can fine-tune these movements and then prevent this motor error. So that will ensure smooth, balanced motion during activities like walking and reaching. In terms of anatomy of the cerebellum, I mean, if you remember, it is here, just below the occipital lobe and posterior to the brainstem. So if you see it from posterior view, something that you can see, it is that it has these lobes that are called the posterior lobes, and then it has this anterior lobe, and then in between the left and the right hemispheres, which is similar to our brain, it has hemisphere left and right, we have a little protrusion that is called vermis. And it's called vermis because vermis means worm, so it looks like a little worm. And then similar to our brain, the surface, it has like protrusions that they follow like a lateral pattern like this, but you're looking, of course, this posterior view. So these ones are called folia. And in between the folia, we have sulci. This folia increases the surface area, just as our cerebral cortex, to keep more neurons. So what are the neurons? Will be the Purkinje cells. And if you remember, I told you these are multipolar neurons that receives around 200,000 synapses, each of them. So this one, basically, it's a highly specialized organ in our body that receives a lot of information. And then deep within this region, we have the deep cerebellar nuclei. And then if you see the anterior view, you can see other lobes like these ones that are called the flocculonodular lobes. And then connections to the brainstem and to the brain that are called the cerebellar peduncles, through which you have the axons of these Purkinje cells, which you can see in this cut. So this is like the parasagittal section, and this is the midsagittal section. So it has white matter, the cerebellum, just as our brain. So this white matter migrates the axons, myelinated axons, through these cerebellar peduncles. that connects into the brainstem. In this case, will be the pons. And then this surface, which will be the cerebellar cortex, will have the Purkinje fibers. And now the name of this white matter in the cerebellum is called arbor vitae, tree of life. It's like that tree that looks like this foliated region. So this is the arbor vitae. So one way to test, before we talk about the limbic system, one way to test the cerebellum, if someone has damage to the cerebellum, is to ask the person to walk extending their arms to the side and put their heel in front of the toes. If the person falls like that, or, well, not falls, but like wiggle, that's an indication of damage to the cerebellum. Or itself, the person will walk like if he was a drunk person. Like you would have ataxia, that's the name of this type of walk. But it's not drunk, the person is not drunk. And then another way is to ask the person to extend the arms and to touch the tip of the nose with their index. We're supposed to have with both hands, so we're supposed to be able to do this without a problem, even with closed eyes. Well, for people who have cerebral problems, they would have intention tremor. So first they will start shaking while they're trying to reach the tip of the nose, and they don't reach it at first. With several attempts, they eventually get closer and closer, and they can touch the tip of the nose. So the bilateral movement, and then be us capable of doing this type of touching of the tip of the nose, is called diadochosynesia. Okay. So now for the limbic system. The limbic system, it is a system that is important for controlling anger, sexual drive, emotions. And it is composed or made out of several structures, including the fornix. The fornix, it is white matter that conveys signals from the limbic system to other brain regions. And then we have the cingulate gyri, which is located in the temporal lobe. It is gray matter cortex involved in memory formation, emotional regulation, and learning. We have the parahippocampal gyrus, which supports encoding and retrieval of information from the limbic system. So it is found near to the hippocampus, which is basically our storage, our memory storage. So we have this hippocampus that is important for learning and consolidation of memory. And then nearby we have an oval structure, is called the amygdala, which is usually referred as the fear center. Many people who has damage to the amygdala will be kind of this person that takes a lot of risk without fearing the consequences, which is part of it. I mean, I won't say that that's the only responsible. We actually have a brain center here in the prefrontal cortex that also regulates the planning, or kind of knowing if you will suffer or if you have a consequence or not. Like, well, for instance, if you're going to have an exam soon, and then you know that that exam is very important, it's very hard, you're supposed to be able to have this region functioning and telling you, well, you better study or else you will flunk. That, together with this amygdala, will make some people to be kind of aggressive, fearless, and get into trouble or do something. Okay, so it is proven. So here we have then this area that is important for behavioral expression of emotion, especially fear and emotional memory. So here is the different regions of the limbic system. So here we have the limbic lobe. So this is the cingulate gyrus. And then here we have the parahippocampal gyrus. And here is the hippocampus, amygdala, the fear center. And then here is the fornix. So this one is the one that relays information to other regions of the brain. So that we can control emotion. So an example, you may have memory about a particular smell in the home of someone, like your grandma, your parents, that brings you specific memories. Like maybe you went there and they have your favorite dish cooked for you, and then whenever you smell something similar again, it brings you back to childhood. Better example, when I was in middle school, there was this guy that apparently liked me, but I didn't like him. It was kind of that time where we all not look our best, so including him. But my gosh, he wear a cologne that smells pretty good. I always give that example, right, Matt? And then, although I didn't like him, and just by him being next to me, I liked to be next to him. Time passed by, of course, we all go our separate ways. We never find out about each other. But then I remember that I asked him in middle school, okay, what is the name of that cologne? And then he told me the name. And then one day I was at this shop, and they had fragrances, and then I asked, well, do you have this? And then, yeah, and they give me like a little card with the spray on it, and I smell it, and I said, this is this guy. So, and then I saw him years after, and my God, he's changed, and I was like, you never know, but okay. Hopefully he didn't say it back like, I was right. But anyway, so that's the limbic system. It brings you back to memories. Okay, a specific time, places, persons. So basically you can tell when a person is nearby because it gives you memories of particular smell or something like that. Now for the functions of the hippocampus. So we have a storage of memory. So when it comes to memory, you have immediate memory. So that lasts several seconds. And then you have short-term memory, which typically can last several minutes. And then that memory, short-term or immediate, can be stored as long-term, or they can go and be forgotten, depending on how relevant is that information for you. Now, if you will go to long-term, it will be actually stored in the hippocampus. So you have to go through the process of what is called consolidation, which is basically enhanced by association with other stimuli. So, that's why here in this course, we use more than one modality. It's not only listening to lecture and all that, but you have to maybe go to the lab and then use the models that helps you, that provides you tactile sensation that can help you to consolidate the memory, repetition, or association. That's another way that you can consolidate your memory. We all know our learning styles, and then that will help. Emotion as well uh, can can help you to develop particular memory. Uh, Sometimes we are very visceral and as persons, and then uh, we had a close friendship, but something happened with our friend that he does something to us, and then we keep that emotion so strongly to us, and then that consolidate that. You say that you forgive, but you didn't forget. So that basically goes to long-term memory, and then you lost your friendship. And then you associate that particular event to whatever happened before. That's why we keep repeating the same story of horror, whatever people did to us over and over again. And then our next friend was like, yeah, I already hear that. I want to hear it again. You move on. That's how we are. Anyway, so that's the hippocampus. Now, basal nuclei. So these will be nuclei around the brainstem that help us to control body movements for the most part, and help us also to regulate the initiation of these movements. The major components of the basal nuclei will be caudate, nucleus, putamen, and globus pallidus. So they send information to the cerebral cortex and also to the substantia nigra, which is found in the midbrain. Substantia nigra, it is an area of the midbrain where we produce dopamine. Dopamine helps us to control body movements. And then some persons might have damage to this substantia nigra. They don't produce enough dopamine, and they have Parkinson's disease, which is basically this disease where people have uncontrolled body movements, and then they have other problems. So basically, this region helps us to maintain smooth motor control together with the cerebellum. That's why they have feedback, all of these. So here is the basal nuclei. So here we have the globus pallidus, and then this is called the corpus striatum. So you have caudate nucleus here, and then putamen in here. All these three form the basal nuclei. Hypothalamus. It is located in the region below the thalamus. Remember, hypo means low or below. And then it acts as a control center for homeostasis, regulating body temperature, hunger, thirst, and circadian rhythm. We have in that region osmoreceptors that can detect changes in the osmolarity of our blood. Our blood has to have around 270 millismoles per liter, and then that increases when someone ingests a lot of salt or sugar, and that makes our osmoreceptors to dehydrate, and then they stimulate the thirst center so that we can drink water so that we don't dehydrate. Also, we have control centers for ANS functions like controlling the heart rate, blood pressure, digestion, and breathing. Also, we produce two hormones, ADH and oxytocin. ADH, it is called antidiuretic hormone or vasopressin. So this one, it is released when someone has low blood volume. If you have low blood volume, your blood pressure is going to go down. You want to preserve as much blood, as much volume in your body, as much water. That one, you do so by releasing ADH, which prevents you from going and peeing a lot. And then oxytocin, it has different functions. In the case of females, it is important for contraction of the uterus during childbirth and contraction of some muscle cells that are located around the ductules for our breast tissue when we are lactating. So when these smooth muscle cells contract under the influence of oxytocin, milk get ejected. And in males, it helps for the contraction of the prostate gland during ejaculation. And also in females during sexual intercourse, it allows the contraction of the uterus. Now, in both genders, there has been some studies that shows that oxytocin is a hormone for bonding. People who are generous and who are very friendly, they have higher levels of oxytocin than someone who's stingy and who doesn't have a lot of friends. So next time, I guess, if someone doesn't give you some gift bag, or, well, not that you want the gift bag, right? But if he gives you like a tiny gift in comparison to yours, you can tell, well, you don't have as much oxytocin, do you? No, don't say that. That's crazy. And then another thing that you have in the hypothalamus is centers for integrating emotional responses. And then since you produce, you have a relay center there for sending information into your pineal gland, it helps you also to regulate sleep-wake cycle and hormone secretion. This is the inferior view of the hypothalamus, showing you some of these centers. So this is anterior, this is posterior. So this is the anterior nucleus. Here we have white matter in between some of these centers. So we have the lateral geniculate nucleus, medial geniculate nucleus, pulvinar, reticular nucleus, that it doesn't have a specific function rather than connecting the reticular formation. We will see in a little bit what is that. We have ventral posterior nucleus, lateral dorsal, lateral posterior, etc. So the functions are in here. Some of them, like in the case of this medial geniculate nucleus, helps to relay information like auditory information. Lateral geniculate, this one, helps to relay visual information. So we will see in a little bit until we see the spatial senses chapter, we will see a little bit more of these two nucleus. And then this is the medial view, showing you more nucleus, including this one, supraoptic, that helps to produce ADH and oxytocin. And then some of these, like supramammillary, that promotes wakefulness, similar to this tuberal mammillary nucleus. So sometimes people might have a stroke in this area, and they have damage to these centers, so they remain in coma, the person, or they fall into coma. Other ones, like this arcuate, helps to regulate feeding. So some people might have damage to this, and they have problems to being basically satisfied with food. This one The dorsolateral preoptic controls the body temperature and promotes sleep. Same for the preoptic medium. So this one regulates body temperature. And then others, like the medial preoptic, it is in charge of regulating sexual behavior. So, I mean, you can see all of the functions in there. So it's a tiny region with many different functions, as you can see. Okay, any questions before we move into the brainstem? Okay, so the brainstem is made by three portions from superior to inferior. The most superior one will be midbrain, then you have the pons, and then you have the medulla oblongata. Midbrain, it helps to process visual and auditory information, carries motor fibers from the cerebral cortex down to the spinal cord, and then works with the basal nuclei, like the substantia nigra, to regulate body movement. And as well, mediate reflexes related to sight and sound. The pons, it is located below the midbrain. It has like a tiny protrusion in the anterior view, so it looks like if it was a walnut. It regulates breathing patterns, so we have the pontine nuclei there that helps to regulate the amount of time that happens between each of our breaths that we take. And then it helps as well to control sleep-wake cycle, because we have a series of neurons that runs between the medulla oblongata and the midbrain that is called the reticular ascending formation that basically keep us awake. And then it helps us relay center between the cerebrum and the cerebellum through these peduncles that we have. Below the pons, we have the medulla oblongata that it has several centers that controls vital functions like blood pressure, like heart rate, breathing. So we have the cardiovascular centers there, all our autonomic nervous system functions. We even have the vomiting center within this region. And then we have motor and sensory pathways, critical for survival, including the reticular formation. So what are the tracks of white matter within the brainstem? So we have the centers that, the tracks that brings information between the brain and the spinal cord, like the corticospinal pathway. And then we have some pathways that relay sensory information from the spinal cord to the brain. So here is the different regions of the brainstem. So here is the midbrain, pons, and medulla oblongata. So this is the anterior view. So here in the midbrain, we have laterally the substantia nigra, and medially here, we have these superior and inferior colliculi of the corpora quadrigemina. Superior colliculi and inferior colliculi receive visual and auditory information so that we can have reflexes accordingly. So, for instance, if someone will be exposed to a sudden strong bright light in their eyes, one of the reflexes, it goes through these midbrain regions, or the stimuli, will go from the eyes into this superior and inferior colliculi, so that we can respond accordingly. So instead of staring at that source of bright light, you're supposed to first close your eyes and then do this movement, turn away from the source of bright light to protect your retina. Another reflex that we create with these two centers, the superior and inferior colliculi, it is when someone hears a loud sound. Let's say that you're walking on the sidewalk, and then you hear, let's say that the houses or businesses are on the right side, and then you hear a loud screeching of tires on your left side that apparently is coming onto you. So with that reflex, you're supposed to turn to the left and look to see if a car is going to go into the sidewalk and hit you. And that way you can respond. Or more likely, let's say you're walking in the sidewalk and then you're alone, and then you hear a dog that is barking at you that is coming closer to you. If it's on your left, you're supposed to turn to the left. If it's to the right, you're supposed to turn to the right. So that is protective reflex, according to that auditory information. Okay? So that's what we process in this area, in the superior and inferior colliculi. And then here we have the pons. In the pons, we have nuclei for cranial nerves. So we will see later on what are these cranial nerve nuclei, but we have these cerebellar peduncles that receive information from the cerebellum into the pons, so the pons can relay it into the cerebral cortex and into the basal nuclei, so that we can control our body movements. And also we have within the pons this pontine nuclei that helps to control breathing pattern. And then we have here below the pons the medulla oblongata, where we have several nuclei, including the inferior olivary nucleus, and then we have the nucleus cuneatus, and then nucleus gracilis, that receives information from our posterior muscles. And then we have within this region the cardiovascular centers that controls blood pressure, heart rate, and then we also have nucleus that helps to control the rate of respiration. And now in between these midbrains and the pons and the medulla oblongata, in the left and the right side, we have these neurons, these actions of the neurons from the pyramidal system, the corticospinal system, that helps to control the body movements. And as well, we have the reticular formation, this system of neurons that help us to stay awake. So functions of the reticular formation will be regulate sleep and arousal, modulate pain transmission and mood, and also maintain homeostasis of various body functions. If someone has a whiplash accident and damages the brainstem, typically the person can break the axon pathways, and then the person will be in coma. And then coma will be this state where the person won't be able to be awakened by a normal stimuli. So one way to know is basically shout or elicit the painful stimuli in the person by rubbing with the knuckles the sternum or pinching here the trapezius. If the person doesn't even make, because maybe the person won't be able to communicate verbally, but you can tell when the person feels pain by like the expression, facial expression, by the person doesn't make a facial expression of pain, that means the person is in coma. So here is the reticular formation then, located here in the brainstem. So it is activated by sensory stimuli, by the limbic system, and the cerebral cortex. So that's what it keeps us awake and alert. Any questions? Now, for protection of the brain, we have three membranes, the cranial meninges. From outer to inner, from external to internal, we have dura mater, arachnoid, and pia mater. They are different. The most external one, the dura mater, it is thicker. It's made out of collagen fibers. It has two layers. One that it is fused to the periosteum, which is the periosteal layer, and the other one that follows the contours of the brain, which is the meningeal layer. And in the longitudinal fissure, that dura mater goes internally, deep into this, and it forms a folding that protects the blood vessels in the brain, that drains the brain of our blood, that are called the dural sinuses in the longitudinal fissure. Now, the arachnoid matter is called arachnoid because the fibers are kind of randomly oriented between the dura mater and the pia mater. And it makes it look like if it was a spider web. Aracna, araña, in Spanish means spider. So this arachnoid looks like a spider web, so it's made of delicate web-like layer fibers that cushions the brain. And in this arachnoid, in this arachnoid matter, we have tiny protrusions that are called granulations that we use to absorb cerebrospinal fluid, or CSF. And then below the arachnoid, and then next to the pia mater, we have a space that is called the subarachnoid space. This is where we have the circulation of the cerebrospinal fluid, not only in the brain, but also in our spinal cord. So we will see the spinal meninges when we talk about the spinal cord. And then the pia mater is very thin. It's made out of connective tissue, much thinner than the dura mater. And pia means soft, so it's the softest of the meninges. It closely follows the gyri in the sulci, and it looks shiny. So this protects the brain more closely than the dura mater. And then the spaces between the meninges. Above the dura mater, we have a potential space that is called the epidural space. So this epidural space is a potential space because potential means that it can be, but it is not. So something that can fill this epidural space is blood. When someone has trauma or a stroke, this space can be filled with blood. And then we have the subdural space, that it is a potential space between the dura mater and the arachnoid that also can be filled with blood in pathological conditions or fluid. And then subarachnoid space, this is an actual space where we have the CSF, cerebrospinal fluid. This kind of a combined word that between that and accent and then whatever. Cerebrospinal fluid, guys. It's much easier to say CSF. Here it is. Look how it is spelled. And then something that the cerebrospinal fluid does, it is nourishing the neurons and also provides buoyancy. So our brain can be kind of floating and cushioned within the cranial cavity. So here is the view of the cranial meninges. So they remove part of these bones in our brain so we can see. So this is a section of the brain so that you can see in the longitudinal fissure the folding. This is the dura mater making this folding to hold these sinuses, these blood vessels that drain the blood in our brain. So we have these inferior and superior sagittal sinuses that carries this blood from our brain. So this is the most external layer. This is the dura mater with the periosteal and meningeal dura. And then here we have the arachnoid, and then underneath the pia mater. And then the pia mater, what it does is not only protects most closely this cerebral cortex, but also helps to form a pathway where the blood vessels, the arteries and the veins run in the brain tissue. And then here is the dural folds and sinuses. So this is the mid-sagittal view. So we have here what is called the falx cerebri. Falx cerebri attaches into the crista galli of the ethmoid bone anteriorly. So this falx cerebri separates the hemispheres into right and left. And then we have another one, another folding that runs like this horizontally between the brain and the cerebellum, that is called the tentorium cerebelli, that forms like a tent for the cerebellum. And then this is a magnification of this region, so you can see the different meninges. So this is a frontal section. This is the periosteum of the bone, and then here the periosteal layer of the dura mater. This will be the meningeal dura. So above here, in between the periosteum and the periosteal dura, you can have the epidural space. Below, between these two, you have the subdural space. And then here is the arachnoid. It looks like a spider web. So here we have the arachnoid granulations that absorb the excess of cerebrospinal fluid. And then the cerebrospinal fluid will be circulating here. We have some blood vessels in the subdural, in the subarachnoid space. And then we have here the pia mater surrounding and following the contour of the brain. Questions? Now for the ventricles. So the ventricles in our brain are cavities that hold cerebrospinal fluid and circulate the cerebrospinal fluid. And these ventricles are four. They are interconnected by kind of tubules. So we have in each of the hemispheres, we have ventricles. So these will be the lateral ventricles. We have left and right. And then these two ventricles are interconnected by the I would tell you later. Well, these two lateral ventricles are connected into the third ventricle, which is just below these two lateral ventricles. And then the third ventricle will be connected to the fourth ventricle. So the lateral ventricles connect to the third by the interventricular foramina, and then the cerebral aqueduct connects the third to the fourth, and then the fourth connects to the subarachnoid space with the spaces that are called the Luschka and Magendie tubules. And then we have the CSF that it helps for cushioning the brain and then removes the waste as well. So here's the location of the different ventricles. So the lateral ventricles are the largest. They look like inverted C shape, and it has horns. These are the anterior horns, these are the posterior horns and inferior horns. And then we have here the interventricular foramen that communicates the lateral ventricles to the third. The third communicates with the fourth, which is anterior to the cerebellum, posterior to the pons, by the cerebral aqueduct. And then this fourth will send CSF into the subarachnoid space in the brain and into the spinal cord by this ependymal canal or central canal. So sometimes some people may have obstruction of this aqueduct or the interventricular foramen, and then they accumulate CSF, and these ventricles enlarges. This is called hydrocephalus. And then one way to tell in MRIs or CT scans is because you may have enlargement of these horns. It's easy to tell. And then they have to diverge that fluid into the heart or into the peritoneal cavity. The foramens or the tubules that brings the CSF into the subarachnoid space in the brain is called foramen of Luschka and Magendie. So for the circulation of CSF, so CSF is produced by capillaries, which are the tiniest blood vessels that we have inside the ventricles. These are specialized and are called choroid plexuses. And then these choroid plexuses produce certain amount of CSF and then recirculates through the different ventricles from lateral to third to fourth, and then subarachnoid space, ependymal canal, subarachnoid space, back into the subarachnoid space where it is absorbed by the arachnoid granulations, and then into the dural venous sinuses. So again, functions of the CSF is to provide cushioning to the brain, maintain buoyancy, and waste removal and nutrient delivery. So here is how the CSF circulates. This is the third ventricle that is just located below the corpus callosum. Here we have this choroid plexus, so if you magnify it, these are cuboidal cells, simple cuboidal cells that secretes the CSF coming from these capillaries, which is the choroid plexus. So then the fluid goes from this area into here, and then all around. And then lastly, it will be absorbed here in these arachnoid granulations. So from lateral ventricles, this is the lateral ventricle, this is not the third. From lateral ventricles to the third, through the fourth, and then into the subarachnoid space, and then the ependymal canal here. Speaking of capillaries, the capillaries from our brain are the tiniest branches of the middle meningeal artery and the middle cerebral artery and the posterior cerebral artery and anterior cerebral arteries, which are branches, these anterior and middle from the carotid, internal carotid artery, and then the posterior from the vertebral arteries. These tiny capillaries together, if you remember, with the extensions of the astrocytes, forms the BBB, blood-brain barrier. This blood-brain barrier, it forms a functional barrier into which nothing that can harm the brain is supposed to go through. Unfortunately, it's not foolproof. Sometimes people can have injury to this blood-brain barrier and end up with infections in the brain and then damage to the brain. So some molecules can cross easily, like lipid-soluble molecules, like ethanol, for instance. Others require a specific transport. And the function, in general, of this blood-brain barrier is to maintain a stable chemical environment for the brain to function properly. So if you make a section of a typical capillary in our blood, in our circulatory system, these capillaries have the red blood cells, white blood cells, and then they have water and nutrient in the plasma. So they have openings between the cells, which is this, so that you have exiting of all of this. Now, the brain tissue, since it's very delicate, very fragile, you're supposed to have an extra layer of protection, which is given by these extensions of the astrocytes, leading to less or limiting whatever it comes to the brain, limiting the amount of items and what type of items supposed to go into the brain. Now for the spinal meninges, similar to the cranial meninges, we have three: dura mater, arachnoid mater, and pia mater, from external to internal, and then similar in structure. So this is the toughest, this is the softest, and this one, the arachnoid, looks like a spider web. And then the epidural space in the case of the spinal meninges, it is a space where we have fat and blood vessels. In the subdural space, we have a potential space between the dura and the arachnoid, and the subarachnoid space is where we have, of course, the CSF. So this is a cross-section of the spinal cord. So here we have the dura mater, so this is the epidural space where we have fat, so this is the vertebral body, and here we have fat, or well, vertebral body here, and then here we have the arch, vertebral arch, and then here we have the arachnoid, and here we have the pia mater. And this is the white matter of the spinal cord, this is the gray matter, and this is the rootlets that form the spinal nerves. And then if you see here in this anterior view, so we have blood vessels that are held in place by this pia mater that provides, of course, blood supply, which brings oxygen and nutrients to and carry away wastes to the spinal cord. For the anatomy of the spinal cord extends between the foramen magnum and L1 or L2. So that's the length. And then it is protected by the vertebral body, and the arch is located in the vertebral canal, and it has some enlargement depending on the region. So it has the cervical enlargement that has spinal nerves that are going to provide innervation or function to our neck muscles, our neck structures, and also to our upper limbs. And then in the lumbar area, we have also an enlargement that supplies the lower limbs. And then in the thoracic area, we have an enlargement that basically gives innervation to thoracic muscles. And then at the end of the spinal cord, we have extensions of nerves that looks like a horsetail. So this is called in Latin cauda equina. Cauda for tail, equina for horse. So it looks like a horsetail, and that's where we have several spinal nerves that are part of the sacral and lumbar area. So here is the different view of the spinal cord, the posterior view of the spinal cord. So here we have the cervical enlargement, here we have the thoracic enlargement, and here we have the lumbar enlargement. And this will be the cauda equina. Now the tip of the cauda equina is called the filum terminale. That attaches into the coccyx so that you have an inferior point of attachment of the spinal cord. And then here is the rootlets, posterior rootlets of the spinal cord in this thoracic region. Now, if you see single segment of this spinal cord, you can see that the gray matter is subdivided into different horns. So this is the anterior horns, this is the lateral horns, and this is the posterior horns. So this one sends motor information into muscles, like somatic muscles or smooth muscles, like located in the urinary bladder or in digestive viscerae. And then these lateral horns send information from the sympathetic and parasympathetic system. Sympathetic system, more specifically, sympathetic system in the thoracic area. And then the posterior horn receives sensory information from the skin, internal organs. So they enter through this posterior root, which is sensory, these axons. So you have sensory receptors in these organs, then they carry an afferent or arriving signal into the posterior horns, and then you have an interneuron that can connect these neurons from the posterior horns to the anterior horns, and then you can create a reflex. And then this gray matter in the center has this ependymal canal, central canal, where you have flow of CSF, and then you have this white matter that is subdivided into different tracks so that you can send information ascending and descending into the different regions of our body. So what are the major ascending and descending tracks of the spinal cord? So the ascending tract, which are located in posterior columns, are important for sending information about fine touch and proprioception. Proprioception, it is a function that tells us what is the position of the different parts of our body. So you know where your head is right now, where your feet are, in what position, because you have proprioception. So we have proprioceptors that tells us that information. That it is very important for gymnasts. So whenever they do this flipping on the air, they're supposed to fall in their feet into the ground. They use this proprioception information. And then we have a tract that brings information about pain and temperature, which is called the spinothalamic. We have a subdivision of this lateral, for instance, and anterior, spinothalamic. Descending tracts bring information from the cerebral cortex or the cerebellum into the different muscles. So we have the corticospinal that controls voluntary movement, and then we have other motor pathways. So here is the section of the spinal cord showing you ascending or sensory pathways and descending motor. So ascending send information from the spinal cord into the brain, and then descending from the brain into the different muscles in our body. So this is motor, this is sensory. So here we have this pathway that is called the lateral funiculus. And then this one that is located in between the dorsal horns is called the posterior funiculus. And then in here we have the anterior funiculum, just located near to this indentation between the left and the right side of the spinal cord. So what do we have here in this posterior funiculus? In the middle, we have this fasciculus gracilis. And then here we have the fasciculus cuneatus. So fasciculus gracilis carries somatosensory information about touch, vibration, and proprioception from the lower limbs. And this one does the same, but from the upper limb and also the trunk in the neck. So cuneatus is for upper limb, trunk and neck, while this one is for the lower limb. And again, just because we have here this medially, that means that this information from the lower limb is more important than from the upper limb. And then here, this part of the anterior funiculi, and then these are called the anterolateral system, spinothalamic tract. So they bring information about pain, temperature, and touch. And then this will be the spinocerebellar tract that carry information into the cerebellum. And the descending tract, we have the corticospinal, these two, that carry motor information into our voluntary muscles, reticulospinal, that carry information into the brainstem, tectospinal, into the superior colliculus of the brainstem. So this is for movement of our eye muscles and movement of our head, and vestibulospinal tract that help us to control equilibrium and maintaining it. So here we have a view of these pathways. So we have first order neurons, second order neurons for relay, and third order neurons in case that you want to send sensory information from the ascending pathways. When it comes to descending, we have two neurons only. First order neuron from the cerebral cortex into the spinal cord, and second order here from the spinal cord into the target. So this is called upper motor neuron, and this is called lower motor neuron, and they cross. So the left side controls the right controls the left. We will leave it in here. Any questions?